Let's look at the layers of the visual cortex now. So, layers of the visual cortex. Layers of the visual cortex. So, when you're looking at layers of the visual cortex, you need to take note that from our previous discussion, we say that we have optic radiations which come from the lateral nucleus to the primary visual cortex, which is found within the calcaline fissure. So this primary visual cortex, don't forget that it can also be called the striate cortex, can also be called the Brodmann area 17, get? So it has a total of 6 layers, just like the lateral nucleus, which we have just looked at. So it also has 6 layers. However, most of these optic radiations, which we also call the geniculocalcaline tracts, will terminate in layer 4. So if you look at this, where do we have our layer 4? Layer 4 is here. But you can easily tell that the way I've drawn these layers, you can see that layer 4 is a bit unique in that for it has divisions which are in black. So I want to tell you that layer 4 has about 4 divisions. It's divided into 4A, 4B, 4C, and then this layer 4C is further divided into 2, that is alpha and beta. You get? So these are the 4 divisions of layer 4, and you find that all the optic radiations will terminate into layer 4. You get? So it will be the same thing this side. So what you need is to follow. So we remember that we had P cells, we had M cells, and we had K cells. So mainly we shall have these two. I think I'll need to organize this clearly. So this side I'll put the M cells, then the other side I'll put the P cells. So where are they going to project, or where are they going to stop? So you need to see that, or remember that all the M geniculocalcaline tracts will move up to layer 4C alpha. You get? So this is where they will terminate. You get that point? So all these tracts will terminate in layer 4C alpha. You can draw it and draw it better in your book or pamphlets, whatever you're using to follow. Then from layer 4C alpha, they can then spread into other layers. You get? They can then spread into other layers. By their termination is layer 4C alpha. That's why you'll find their main nucleus of termination, layers 4C alpha. And remember, we say that the M cells, they have information concerning with motion, black and white, it's slow, and it's mainly from the rod cells. Then when you look at the P cells, for them they will terminate in two regions, but mainly they will move into layer 4C beta. You get? So they will move into layer 4C beta. So if this is our alpha and this is beta, they will terminate mainly in layer 4C beta. You get? So they terminate mainly in layer 4C beta. So from layer 4C beta, some of them will terminate in layer 4A here. So we shall have another nucleus here in layer 4A, then others in 4C beta, like that. Then of course from here we can have them spread into other layers. So how about the case cells? So the case cells, these ones, according to your books, they will tell you that for them they terminate mainly in layer 1 and 2. However, some of them can go to layer 4A first, then spread into, project into layer 1 and 2 mainly. You get the point? So they project into layer 1 and 2 mainly. So that's where we have the case cells within the visual cortex. So that's the arrangement of the visual cortex. Talked about the 4C alpha for M cells, then 4C beta for P cells. That is P cells concerned with the fine detail and color vision, which are slow conducting. M cells are rapid conducting. Then the K cells for blue, yellow, color vision. Then other types of cells which we need to look at that are found within the visual cortex, we need to cover them. We have simple cells, we have complex cells, and we have hypercomplex cells. Hypercomplex cells. So, how do these function? I'll just go straight to the point that simple cells, if I'm to draw a picture, let's say I'm going to draw a tree. I start with drawing maybe some lines like that, you get? I draw some lines, so that's the importance of these simple cells. They're just for detecting lines and edges. Lines and edges. You get? That's for simple cells. Then the complex cells, for them, they will care about the orientation of these lines. Let's say whether it's horizontal or vertical. So these are for orientation of the lines. Orientation of the lines. Then how about the hypercomplex? Hypercomplex, for them, they will determine the length of the particular line. Length of the particular of the line. You get? So you'll find that they can also be called the end-stopped cells. End-stopped cells. I'll explain why they are called so. End-stopped cells. So why are they called the end-stopped cells? You find that if I'm drawing a line and my mind knows it that maybe this is has to be 4 centimeters. So the moment I perceive that they are already 4 centimeters, the hypercomplex cells will be excited by what? By the complex cells. The complex cells will excite the hypercomplex, telling them that now you know what? This is already 4 centimeters. However, if the 4 centimeters are exceeded, you will find that again we shall have inhibitory signals from the complex to the hypercomplex, and this is what we call the end-stopped concept. In that, if we exceed the given length, we shall have them inhibited. That's where the end-stopped name comes from. So those are the other cells that we have in the visual cortex, and at this point, I believe we have covered what we are supposed to know about the visual cortex. The other one, which I'll talk about briefly, is the secondary association cortex, which I talked about earlier on. I said we have the dorsal stream, which was going to the parietal lobe, and then we had the ventral stream, which was going to the temporal lobe. We said that the dorsal stream was mainly for motion and spatial processing. Then the ventral stream is mainly for perception and recognition of objects. You get? So don't forget that. And with that said, I believe we are done with the visual cortex, and we can proceed to the next part.